So we'll be moving on to our next part right now. And like I said, if you have any questions, go ahead and send them off to me. So Trink, I'm going to throw this over to you now.
Hi all, I'm Trink Praxel, and I am one of the members of the Public Education Committee for our local UC Master Gardeners in Monterey and Santa Cruz County. So we love to put together these programs for you, and we're really lucky today to have two of the primary leads for our propagation team. And it's particularly good to have them because they're going into a spring sale very soon. So they're very busy. They have been propagating all kinds of wonderful plants for the sale. And I think you'll really enjoy getting some of their techniques down. I did want to point out that we are calling this a basics class. And our intent is later in spring, sometime early summer, soon, we'll have a more advanced class. And you'll hear Diane and Charlene mentioned a couple times, this is something we're going to cover in the next class. So just that's what they're referring to. So when I say this first class, the one today, and then the hands-on experience on Saturday, and you're going to hear a couple of us again say, we don't have many of you registered yet for the Saturday class. We would very much encourage you to come to that because you're going to actually be able to get your hands dirty. And you know, there's nothing for a gardener better than to actually do the things with your hands rather than just have the knowledge in your head. So it's supposed to be a nice day on Saturday, no rain until much later in the day. So do consider that. So who are we? First of all, I just want to make sure, I think most of you know this if you've been to our classes before, that the UC Cooperative Extension Master Gardener Program, we're just one element of it here in Monterey and Santa Cruz County, and we extend research-based knowledge that comes out of the UC system. They're wonderful faculty. We're trained, but with their information, we have resources from the University of California that we can access when needed to answer questions. And so we're here, that's our role, to get that UC information out to the home gardener on pest management, sustainable landscape practices, etc. So I hope you enjoyed this class, and we're gonna and forgive me, I'm controlling slides today, and occasionally they get a little hard to move. So sorry, but see that, there we go. Now Diane and Charlene are gonna introduce themselves. Go ahead.
Hi, I'm Diane Apostolos, and I am a Master Gardener. I graduated in the 2020 class. I am currently part of the lead team of the Master Gardener propagation team. We're the ones who produce the plants for their sales. And my passion is landscaping for wildlife. And right now I'm really doing a lot of work trying to grow my knowledge about all things plants. So that's where I am.
Charlene?
I'm Charlene McCowan, the Master Gardener class of 2020, a classmate of Diane's. And as she mentioned, we run the plant propagation project, which produces the plant sales for the Master Gardeners. But this is our first online class venture. So you are our guinea pigs today. And I put retired educator on my introduction slide here to remind me to just mention something. I was a high school principal and high school English and Spanish teacher for many years before I retired, but I wanted to mention that nothing in that part of my career ever quite matched the wonder and the curiosity that I saw in students when we were working in the garden, in the school garden. And something about digging up purple potatoes kind of reduces any cool teenager to an 8-year-old in terms of enthusiasm. And I get that. And after all these years, propagation is still unfailingly miraculous to me, whether it's from cuttings or seeds. So I think that's what we're kind of hoping to impart to you today. We are very passionate about this, and thanks for coming. Diane, I think you're on.
I'm on, okay. So I'm just going to let you know a little bit about how these slides are organized so that they make sense to you, not as you're going through them necessarily, but maybe as they're finished. So first, I am going to be presenting about how to prepare, root, and care for herbaceous cuttings. But I want you to know there's some fundamental things to propagation that will make it very useful for you to make decisions about what kinds of plant material to cut. And so I'm going to go over a little bit of plant morphology to share that. And then we're going to get to selecting planting materials. And then finally, how we're going to stick, care for, and finally plant the rooted cutting. So I think I should tell you now, because I know this is probably not going to, it's going to not make sense, but I oftentimes will use medium and substrate, and they mean pretty much the same thing, except when I use substrate, it's generally when there's a plant in the medium that's, acting as a substrate for the plant. And then plant, parent or mother plant, and both of those are going to mean the same thing. And then I use the term sticking because when you have a cutting, there's not much to do with it but stick it into the medium. So that's the term that we usually use. So it's kind of unusual if you're not used to hearing that term. So, okay, to begin with, there's two types of propagation. One is sexual, and that's the union of sperm and pollen with egg in the female plant to create a seed. And you have on the side here a little petri dish full of Rudbeckia hirta, and that was to look at the germination process, which normally we don't see. And Charlene's going to get into that and talk about what normally, when you read on a packet, what that means. And then asexual propagation is basically making a clone of the parent plant using either leaf cutting, stem cutting, root cutting, and other different types of strategies for manipulating the plants. And one of the reasons that we do the cloning of plants is that some characteristics do not translate through seeds. The plant on the right-hand side here, which is Plectranthus ciliatus, it's gold. Its leaves are very unique. They're maroon underneath, and they often are lime green on the top when they're in the sun with little red streaks in them. So those are the characteristics you want to look for and only be able to capture with a cutting. Okay. Okay. So there are advantages to asexual propagation, as I said. Physical characteristics, sometimes those are growth habits, the color of a flower, or as I said, the foliage. And biochemical characteristics typically are reproduced to have a bit more disease resistance for plants that would be more susceptible to specific diseases. And then most of you probably know that with hybrids and cultivars, seeds don't always come true to the mother plant. So to be sure that you get the same type of plant, you would want to reproduce that with vegetative cutting. And then some plants are actually sterile. They don't produce any seed or they don't produce any viable seed. So you'd have to use plant material to reproduce those. And if you want a specific sex, I think of Garryoleptica, the tree that has very long silk tassels. And that basically horticulturally used is just the male of that tree because that's the gender that's most attractive in the garden. And then juvenility. Juvenility is before a plant reaches its maturity when it can produce seed and produce fruit, which is where the seeds would come from. So you would want to use a plant in its mature state to be able to bypass juvenility through the growth process of the new plant. And then faster production time, if you know that for us, for instance, the spring sale is kind of a challenge because the winter is not a great time for growing a lot of plants. It's not their normal growth period mainly. So it does cut down on the time frame to get to a marketable or a plantable size. And then there's another thing that just escaped me. Oh, some plants actually take years and years and years and years and years, like decades, to reach maturity to flower. So those plants typically, to have enough in the marketplace, I suppose, are reproduced vegetatively as well. So thanks, Trey. And then the disadvantages, of course, are the loss of genetic diversity. And that makes plants more vulnerable to disease, and then also we can't produce new varieties because there's no different genetics to mix together to create a variety. And the adaptability to environmental changes they would not have any ability to, since there's not a generation that happens fast after one another, to adapt to the environmental conditions. So if you have patented plants that are not going to change for 20 years, you're going to have a plant that's not going to be able to maybe do that well 20 years from now based on the environmental conditions. So what is a vegetative cutting? A cutting is any part of a plant that's taken from the parent plant and then treated in ways that stimulate regeneration of missing organs. So leaves, stems, and roots can serve as cuttings. In this section, we'll focus on the techniques used to propagate herbaceous stem cuttings. I just wanted to include this little picture on this side of this root coming out of, what is the name of this plant? This is a, can't remember the name of the plant, but it has very, very small little sections in it, and a root can grow out of any small section in there. And this particular root has gone beyond just the single radical root that comes out and has its filials starting. So just wanted to give you a picture of what you won't see when you're doing a cutting, because that will be below the substrate of the medium. Okay, so here we go into a little bit of plant morphology. Meristems are really important for reproduction, vegetative reproduction. And these are areas in a plant that are actively growing. I mean, the cells are actively dividing, and at some point they can undifferentiate. Okay. And then they will become cells that will form into organs that are missing. And in this case, we're looking for cells that will hopefully regenerate or redifferentiate into root cells. So this process is called totenpotency. If you think about humans, you know, when we have, say, for instance, we have a liver cell, it's always going to be a liver cell. No matter what we do, it's going to be a liver cell. With plants in these meristematic areas, the plants they're already designed to be something. They can undo that and then redesign themselves to become another organ. So we're looking to get adventitious roots from these meristematic areas. And an adventitious root is a root that grows from any non-root tissue. So those are just some principles that you want to know about when you're thinking about how to select planting material for propagules. So
I think this is just one of the most amazing parts of propagation, and to just know this about plants, you know, that the meristem there is just actively dividing and it's undifferentiated and just constantly creating new material. It's no wonder that we can reproduce them in the ways that we can. It's really... Diane, would you explain what we said in the previous slide about herbaceous cuttings? Just make sure that we all understand that.
Sure. So I'm going to explain it later in other slides as well. But an herbaceous cutting is basically a cutting from an herbaceous annual or perennial. So the material is very easy to root typically. And it's a good place to start for being, you know, to do your first cutting, because you'll have a little bit of success.
Great.
So thank you. Yeah.
Mm-hmm.
Okay. So some of the things you want to be aware of when you're selecting planting material is the seasonality of a plant. So there's genetic coding for when a plant is in active growth state. And you want to pick a time when that particular plant is growing. And pretty much for herbaceous plants, we're in a very good time of year to grow those, because they are now just starting to move into their growth time. Other plants might be growing in the fall, or the winter might be their growth cycle. So you have to have an awareness of when their growth cycle is, when they flower, and choose before they flower, but while they're growing. And then it's also very important to look at the age of the plant and the health of the plant. We don't have to worry about that too much in this class because with herbaceous cuttings, you're going to be dealing with annuals and perennials, and they're typically they're much more short-lived than shrubs, trees, and subshrubs. So you want to make sure that the plant is healthy, well hydrated, well nourished, and free of pests and disease. So when you think about what plant you might want to use for a cutting, you want to look at it a few weeks before you would take the cuttings. And if it looks like it needs more water, or it looks like it's the leaves are a little bit faded, you might want to fertilize it and water it and keep an eye on it until it looks like it's very hydrated and healthy and ready to go. And then I want to talk a little bit about plant parts, because those are also important when you're going to select a cutting. So Trink, can you put your little pointer in there? We'll talk first about nodes. So a node is a part on the stem where a leaf attaches. So you'll have either a leaf or a former leaf that's fallen off, and that will be a node. And then you have, well, here's the terminal bud at the very top. So, you know, that's another growing point. And there's an axillary bud that sits on the top of the petiole. You can actually see one there. This one is kind of doesn't look so much like a bud, but it is. And that is also a very,
oh, here, yeah.
There and up here on the next one, you can see the bud sitting in there.
Yeah.
So that's called an axillary bud. And then conceptually, you need to know about the inner node, and that's basically the length of space that's between each node. So that'll come up in another slide, but these are all important parts of the plant that you want to be aware of for knowing that they're going to grow full of the potential to develop oh gosh, it's a meristematic area and can ready to grow the adventitious roots. Thank you. Take a breath. Okay, so there's a couple of ways to classify stem tissue, and when you're classifying mature stem tissue, you're going to have herbaceous materials, and they come again from annuals and perennials. And then there's hardwood, semi-hardwood, and softwood cuttings. And these come from woody trees, shrubs, and subshrubs. Yeah. And the techniques for each of these is different, very different. And so we would be looking at the hardwood, semi-hardwood, and softwoods for our the next session. So start easy and then get complicated. And then the other way to classify stem cuttings is which part of the stem you're going to be using for the rooting. The tip cutting is always where you have the apical growth area. And then, I'm sorry, the terminal bud. And then there's a second cutting, which is a nodal cutting. And that would be, this is the same piece of material that's cut just a few, like what do I have, four or five maybe nodes on there. So this one would be the nodal cutting, and this one is the stem cutting.
Yep.
I'm sorry, the terminal cutting.
Terminal cutting, yep.
The tip cutting, tip.
And this is the nodal, because it's not really, it's not the tip of, this is the whole branch, this is right. the branch. And so once you cut it, you've got the tip here at the top of this particular cutting, but you still have another cutting with lots of nodes that will also be able to reproduce. Yeah. Okay.
Okay, to prepare your cutting medium, you're going to first want to know ahead of time how many cuttings and what the size of the cuttings are going to be. You're going to want to know a depth, and that would come into consideration when you're looking at the inner node area, because you would like to have at least two nodes under the surface without the bottom of the cutting touching the bottom of the tray that it's in. So if you're going to have fairly long areas that are going to be under the substrate, you're going to want to use a deep sort of container. And then for doing production cutting, you're going to want to use probably a nursery tray. So the second picture is in cutting a nursery tray. And just to let you know, sometimes people wonder how you keep just perlite and peat moss in one of those containers. You choose a container that has very small, like I think this is about 1/8 of an inch hole, a square hole in there. And some of the perlite comes through because obviously it's a little bit smaller than other pieces, but this would be a suitable tray to use if you're going to select having a lot of cuttings. And our medium or substrate recipe is 13 parts of perlite to 1 part of either peat moss or coir. The perlite creates a great deal of aeration and the peat moss holds moisture. And then you would water it and compact it, and then it's going to compact, and then you compact it with maybe a 2x4 that's clean and just press it down so it gets pretty firm. And most of the time you'll have to put in a new layer if you want to get it up to the edge of the surface and press it down again. So it's a process of just, getting it in there and stable. Okay. So preparing your cuttings, the first thing you want to do, you want to get prepared ahead of time because you want the process to go by fairly fast when you're working with plant tissue that loses its moisture pretty fast. So you're going to want to work in the shade or indoors. First, you're going to cut outside, but if you're cutting outside, you're going to probably, depending on how long it's going to take before you get in the house to do the cuttings or in the shade, you might want to put it in an ice chest with some kind of cooling in there to keep it from wilting right away. You're going to work on a clean surface. You're going to disinfect your clippers. And most of the time when we work with, I'm going to say preparing propagules, which is what we call a cutting that's ready to be propagated, I would disinfect that tray that you're going to put the prepared cuttings in, and always keep a spray bottle. You want to mist the cuttings as they're being worked with, but you don't want them to get wet or sit in water. The plant material, you're going to want to select. Okay, so here we have on the side, this is a diplacus, and this is our ribes.
Ribes down here?
Yeah.
Diplacus.
Diplacus, yeah. And you want to select active growth. So if you look at the diplacus on the top, you see that it's very, very green at the very top. And that's kind of a wobbly area on a plant. So you're going to go down to the earlier this year's growth, which is a little bit more stable, and select this portion to be under the ground or the sticking part so it can, doesn't bend over and all that. And then down below with the ribes, it's the same thing. You can see all of these have fairly short internodes, and I'm sorry I didn't put anything on here so you can have perspective, but they are fairly short, probably 1/2 an inch or 2/3 of an inch between these, especially on the fast growing parts, and the same basically on down below. So with the ribes. Short internodes, remember, and keep things well hydrated. Okay, so this is the same piece of material that's prepared for a cutting. So when you cut, trim, and mist, first you want to select sections that are fairly uniform in their size. As you remember, the cutting that I showed you in the glass, that's a 3-foot long cutting. That would probably, fortunately, it roots in water. It would probably not be suitable to put in this type of a substrate, the perlite and peat moss. But most of the cuttings that you'll be using with the herbaceous material now, you can probably get 3 to 5 inch cuttings, and that would be suitable to get a nice plant. So you want them to be fairly uniform in length. You want a minimum of 2 nodes trimmed back with the axillary node can actually remain on the stem. And then when you're thinking about how far you're going to cut it, you want to have the at least the third node up, having the axillary bud on it, and you would like that to be sitting on the top of the substrate, because that will be the first offshoots that come, and your plant will look a lot fuller as it grows, and it's got bottom material on it for growth.
Diane, am I pointing to the right one node here? So this is the third node. So
yes.
This would be above the medium, and this would be below, correct?
Correct.
Okay.
Yeah, and this with the it looks like it's got the shoot. It's a little bit of a bigger, it's a evolved axillary bud. It's already started growing. But, yeah, so that would be where the axillary bud is. And then I also wanted to mention that I reduced the leaf size. Is important because you want to reduce the amount of exit moisture area. So, and then you can't really tell on the ribes, but that has been cut in half. All those leaves have been cut in half.
Oh, yeah.
And then the big deal is for these, you don't have to worry about it because it's very obvious where the top and the bottom of the cutting are. But there are some materials that you're going to be working with, especially with semi-hardwoods, that the insertions of the leaves don't give you any indication of the directionality of the cutting. So you have to be aware when you first start cutting to place the cutting with the top at the top of the place you're looking and the bottom at the bottom. That you won't have much luck if you stick the top in, or if you do, it'll be a very odd thing.
And you cut away any leaves off of the down.
Yes.
Okay, just to be clear.
I just didn't I'm sorry, I didn't articulate. Okay. Okay, and here, this is a lesson learned slide as well. Sometimes you're so directed at preparing the cuttings and getting the plants done, and you don't, oh, I know what plant that is because I did that today. But then you've got 5 different plants in a cutting place, and you can't remember what day you cut it. What the plant really was. Was that a Diplacus with a red flower? Was that a Diplacus with the yellow flower? So it's really, really prudent to remember to do a label at the beginning of when you know you're going to cut a plant. Start there, put the date at the top so you know what month, day, and year, day, month, and year, month, day, and year. Which cut it, yeah. And then the botanical name, because it's just important to know the botanical name, and you can put the common name if you're not familiar with. Just recognize the plant as the botanical name. And then it's good to have the provenance of the material source. So if it's a seed, you want to know where the seed came from. If it's a cutting, you want to know where did that come from? Did it come from the demonstration garden? Did it come from Diane's backyard? Is it a Charlene clipping? Is it whatever? So it's good to know that, just because it is a clone, and it's good to know the provenance. And the other really good part about this is you can use the back part of the tag to track its growth essentially through when the root actually comes in and how many cuttings made it to actually rooting, and then how long it took to grow out in, I'm calling it a rose pot RP, but it's a 2.5 by 2.5 by 3 inch little pot that we usually start the cuttings in. And then you can see how fast that grows out, and you can put it in the next size that you think it will grow into very soon. And you can track how long it takes to get from the beginning of sticking the cutting to when it's a plant that's either saleable or plantable on the ground. And that matters when you do sales of plants. So it's good to have some history, and you won't have it if you don't track it. So
great.
So you can't see my tags in here, but they are in there. I have lots of tags in there. So to get the cutting stuck, there's 2 methods. If you're doing a smaller tray like this, it's just a little bit easier, so you can utilize all the space. They don't always stay in exact rows, but you can just use a skewer, or what do we use? We use a chopstick, yes. We use a chopstick. And you can just poke a hole and put the cutting in the hole, and then just what you would do is just tighten it around the hole to hold it so that the cutting stays upright. Or if you're doing the production style, it's much easier to just cut with a kitchen knife, a trench, and then just put your cuttings in as they come up. It's a lot faster, keeping them about 1 inch apart, and stick them no more than 3/4 of the way into the medium, and try to avoid the leaves touching. And I would say the first thing you want to do before you stick a plant is stick your label in there at the front of it, on the row. So anything I missed? Okay. So for the, when it's a larger amount of cuttings that you're doing and you're actually trenching the medium, this is a good way to come back and stabilize the plants, and we just call it snugging in. So the plants get, they stay upright when they go through the watering process. So it's just a gentle kind of touching and making sure that there's a little bit of compaction and the medium is touching the bottom of the cutting. And then to water in the cuttings, you're going to use a nozzle on the spray mode, on a hose, and it's not going to be a heavy drenching. You're going to want to do like a slight or medium drenching. And first you start by putting the hose straight down, so you don't flip out all the substrate. So you go around the edges first, and then you go in the middle, and then you slightly increase the pressure of the hose as you're watering it in. That way, the substrate gets fully, you have to water it all the way through, so the water's coming out. And the substrate gets really tied up against the cutting so that it holds it in place, and it also makes sure that there's enough moisture for it to retain its rigidity.
And this is another place where seeing it done on Saturday is really useful. It's sometimes talking to these two as developing this class, I can tell you there's certain things that are hard to describe. So seeing it is much better.
Yeah. So you want to place your cuttings in an area that's about 50 to 70% shade, but has no direct sunlight. So I see that more as a quality of shade. It's not like putting it under a canopy that doesn't ever get any light through it. It's more like about half of it is light enough to grow, but it doesn't get sunshine directly on it or a lot of heat. You want to also have an environment for the plant to grow that's high humidity. And so you want to try to place a humidity dome over the tray. I have to tell you that over the big nursery trays, we don't have a humidity dome for that, but I intend to invent one because it seems like it really helps a lot, keeping the plants from drying out too fast and the substrate from drying out too fast. So you're going to want to watch the substrate for the moisture content. And the best way to do that is just stick your finger in right after it's been watered and see what the moisture level is just by touch. And then, throughout the days that you're watching it, you want to do the same thing, stick your finger into the substrate, and you'll be able to tell if it seems like it needs more moisture, and a little bit more water is not going to hurt anything. So, and then housekeeping for this, it's, you do lose some cuttings. Some of them just don't have the vitality to live through the process. It is a pretty traumatic process. And then you have leaf loss and droppage. So you want to keep that clean to avoid any pathogens that could get in there. So that's the other big deal. Oh, and you're talking, Trink, but your mute is on. But I think what you may be trying to say is these are lettuce containers. They mirror each other, but you flip it from one side to the other. This could have a better seal on it, but it is holding humidity, but that's a way to, you know, on the smaller containers, if you can get a matching one to put it on top. And then sometimes if you just do a clip or do a little bit of tape around the side, that's adequate enough to keep it stabilized. So it acts as a humidity dome.
And isn't it great that you can see
yes,
sometimes.
Yes, yeah. You can see the roots growing when they do. That's helpful.
That's great. Okay.
Okay. So how will you know when the roots are there? I would give it I wouldn't start trying to see if it's rooting anytime before 4 weeks. And that would be for some of the faster rooters, which are, you know, the mint family, non-native salvias and teucrians and that type of thing. Then the other ones I would leave for 5 to 12 weeks, and I probably wouldn't touch them for 8 weeks. The California natives, which seem to take longer to root, and then perennials. So when you think that it might be rooted, you're going to do what we call a little tug test. And basically that's to feel and assess the resistance that's coming from the plant roots that might be in the medium. So oftentimes you'll have the same plant that looks exactly the same sitting next to each other. One of them will actually have a lot of resistance to the tug, and the one next to it might not. So, and you'd feel the difference. So if that were the case, I'd probably leave them in for a little while longer till the one that was still fairly loose, maybe stabilized a bit. It's okay to leave them in longer than take them out earlier. So the tug test is a really important thing to get experience with. So again, if you come to the Saturday class, you'll have an opportunity to do the tug test and you'll be able to see the difference and know what to do. However, if you do pull it out and it is not rooted, or if you lose the root when you pull it out, most of the time you can stick it back in and you know, try to get it to go again. Another thing, I don't think you see this so much on herbaceous cuttings, but on some of the other types of cuttings, you'll see a callus at the very bottom, and that's the beginning stage of the roots starting to emerge. So you would want to put that back in and let that grow for a while longer, but know that it's in process. Okay, so I'm going to explain this cutting before I start talking about it, because it's a funny, the note on it is kind of funny. So this is a rooted cutting, and this would have been node number 1 where the cutting happened. This is node number 2, and then this is node number 3 that had the axillary bud on it. Okay. And this was the top of the substrate for the medium. So when you know that tug test wise, that the root is in place and you want to check it out, you're going to slip in next to it with either a chopstick or a kitchen knife, and very carefully just try to loosen up the substrate around the plant, and then by holding onto the top of it very slightly and pushing from the bottom with the chopstick or whatever, you're going to pull it out and check to see what the root looks like. And it's going to be full perlite, and just leave everything there. Don't have to move it off or anything. The best thing you can do is not touch the roots. And then you're going to hold it by the stem and get ready to plant it, which I believe is on the next screen. Okay, so I have my potting mix all mixed up and ready when I have my cutting tray ready to be checked and take out the cuttings that are rooted. So I like to get all these materials again together before we start the process, because it's good to get it out of one substrate into another pretty fast. So depending on the root mass, most of the time these cuttings are taken out and put into a 2.5 by 2.5 by 3 inch little pot. Sometimes people know it as a rose pot. And you want to be able to have a little bit of the fluffy medium, not too moist, but fluffy medium at the bottom of the container, and hold the, where the top is, just hold above that and have that be where you want the top of the planting medium to be. So hold it and then just start dumping, pouring in with your hand the rest of the medium, the soil, and let it be really light and fluffy. And then you can slightly tap it on the bottom of the container to get it settled in. It doesn't have to be snugged in there. Do not push on it or press it in. And then when you water it in, you'll see that the medium will compact slightly from the water. And if you need to, fill it up again to where the top would be and rewater it again. And then let the plant grow out in that size pot until you can tell that the roots are coming out. And you will know by either the roots coming through the bottom, which might be a little long, but also give it a few weeks depending on the weather, and check out the roots. You just kind of slightly tip it up, and you can pull that out because it's going to have some protection around the root system now because it's in the potting soil. And then you can check to see if it looks like it's time to put it in a larger size pot or not. So, and I think I just said what we're going to talk about the next slide. So here's the little rose pot with a cutting that's ready to probably be potted into. I would probably put that in a quart size to let it grow out. You want to, again, check the root mass before you start doing the transplanting. And most of the time you can transplant to a quart, and sometimes you can actually transplant to a gallon because the roots are so robust and the plant is fast growing. And this is true with natives. Once they get their roots growing, they're pretty robust on the bottom, and then they can grow on the top. So,
and do you, sorry, Diane, do you recommend when you're checking the root that you actually like turn it over and take the, look at the root ball?
Sure. So you put two of your fingers in between the plant and the edges of the pot. You're going to turn it over, and that's to stabilize it. And then you're going to just start pushing, just patting a little bit. Yes. And then it'll loosen it up. And then you can, you'll want to let it fall out. You don't want to pull it out. You want it to fall out into your hand. So
great.
Okay. Okay. So Saturday class, we're going to have fun. We're going to prepare the cutting medium and prepare the cutting tray, so you'll be able to see the process and how that goes, and feel the substrate that you need to have in place. You're going to select cuttings from provided material that we'll bring, or if you have some herbaceous annual or whatever material you want to bring, please bring your own. We'll work on that. And then prepare to stick in water in your cuttings, and we'll have a container for you to bring them home in. Dress in layers, wear closed-toed comfortable shoes, bring water, a hat, and small clippers if you have them. And it'll be fun.
And we'll be in probably in a greenhouse out at the Salinas facility. That is our UC A&R office, where for the Monterey and Santa Cruz areas, where many of our farm advisors work as well. So you might see some of them around, but we have the Master Gardeners have our own little space. So it'll be fun. Thank you.
Now I'm going to turn it over to my good friend and propagation buddy, Charlene. She's going to talk about her stuff.
Thanks, Diane. That was great. I know it was mentioned before, but I see a few questions going into the chat. I answered one that was a clarification, but the rest of them we'll take them at the end. So don't worry, you're not being ignored here. So let's talk about seeds, propagation by seed. And we do really hope that you can come on Saturday. The highways are all open if you live in the Santa Cruz area now. And it does make a lot more sense to be doing this hands-on. So today we're going to focus on seeds that have fast germination and fast growth rate. Talking just a little bit about the science behind how seeds work. When a mature seed that's ready to break dormancy is put in a favorable environment, it will germinate, which means begin active growth, develop into a seedling, and then become a mature plant. And I wanted to point out the term germinate gets confused a lot by as a synonym for sprouting, and that's actually inaccurate. The active growth that germination refers to is not something that's observable above the surface of the soil or the medium. And so I don't know, I just felt like pointing that out. It's not a deal breaker, and it does get used incorrectly many times on the back of seed packets. So all right, the seed development, there are three parts of a seed. There's the outer coat, which protects the seed. And then there's the endosperm, which is the food reserve tissue. And then the embryo. And I apologize for the quality of this, but I really like this visual for the top piece here. And I'm going to actually read that part, just the top part to you if you have trouble making out the clarity on your screen. So this is how things begin. Water enters the seed through small openings in the seed coat. The seed then swells and the seed coat ruptures, allowing the entry of gases and the uptake of water. Then as the cells become hydrated, enzymes are activated and begin to break down the storage tissue and transfer nutrients to the embryo. The embryo, that's that top picture there, begins to grow and the seedling starts to emerge. I think the other pictures there that are depicted are what we're, the pieces that we're much more familiar with from our 7th grade science textbook, or, from slides that you see having to do with gardening. The rooting begins, the sprouting unfolds, pops out of the medium surface, etc. But I just wanted to use this sort of as a fulcrum for the rest of the presentation here, that almost everything we're going to cover tonight, talking about optimal conditions for seed germination, is focused on encouraging this process to happen successful. So that's my science lesson for the night. You can see some significant differences in the sizes of these different seeds. It's just a smattering of herbs, flowers, and vegetables. I stuck a dime on there so you could kind of have some perspective there. So what ensures the best chance for seed germination? Quality seeds, proper storage, seed treatment when needed, and that is going to be covered in the next class, not in this one, and then optimal conditions when seeded. So let me define some of this for you. Quality seeds means a couple things. It means that, most importantly, a reputable source. So commercial companies, if you are harvesting seeds or you are using seeds that have been harvested by friends or relatives, the harvest timing and sterile methods are super important. Quality seeds can also refer to the age of seeds. Most commercial seed companies have the dates, usually the year, very clearly labeled on their packets. So I would, especially for beginners, you want to buy new seeds, obviously, and they do generally have a higher germination rate than older ones, but there are plenty of kinds of seeds that actually, tomatoes, for example, they germinate at almost the same rate when they're 7 years old as they do when they're new. So it's not that you should throw away all your old seeds every year, but beginners should definitely purchase new seeds for maximize for success. Proper storage means cool and dry. And the treatment that we're not going to talk about tonight, that would be something like optimizing seed germination by refrigerating it for 5 or 6 weeks before sowing it, or also scratching the surface of the seed coat on some seeds. So we'll talk about that in the next class. Yeah. And then optimal conditions will greatly affect the success of seeding. So you want to pay attention to that. If you are paying attention to that, you can count on 80% of your seeds germinating, I think, and out of that, probably 75% producing sturdy enough seedlings that'll become mature plants. So let's look at how to create those optimal conditions. You have to pay attention to water, oxygen, light, and temperature. Those are the key elements. So for water, seeds have to absorb the water, as the diagram showed, in order to kickstart germination. And then once germinating, they can't dry out or the embryo will die. That's the importance of keeping the medium moist and consistently moist. And then when before the seedling pops up, it's probably easiest to do that by covering it. And I do have a picture of a seeded tray cover coming up later in the presentation. As soon as the seedlings emerge, though, it's important to pull the cover off. And then you embark on the delicate balance of keeping those seedlings moist, but not waterlogged for the best rooting. Oxygen is essential for seed germination also. The respiration rate's low, but some is required. In terms of light, greenhouses and sunny back porches or rooms can do the trick for light. Many gardeners use artificial lights or grow lights. I have resisted doing that myself, but there are plenty of gardeners that do that. And if you're using artificial lights, you want to place them 6 to 12 inches above the seeds. And you also need to keep a watch once the seedlings pop out on the growth, because grow lights can encourage over leggy growth. And you have to pay close attention to the amount of artificial light to allow for normal seedling growth. You're trying to simulate, but not overstimulate. So temperature can't be stressed enough, the importance of paying attention to it, especially with this crazy weather we're having. And for us, of course, working at the greenhouse with the artificial deadline of a plant sale, we're really paying attention to making sure that the plants we want to sell are all going to be ready on a certain date. So home gardeners don't have to worry about that quite in the same way we do. But that said, heating mats are a wonderful help for any seed to germinate quickly and reliably. And all seeds, even the toughest natives, absolutely love warmth in their germination stages. So why not give it to them? Optimal temperature for seed germination is usually somewhere between 65 and 75 degrees. And most seeds do have a minimum and a maximum germination window that's recommended for, their variety. Mats also help ensure success when you're starting seeds indoors, to get a head start on their maturity when it's still winter outside. So that's what we're talking about right now. We have winter conditions, even though it's headed towards spring. And so, for example, with us, we start our solanaceous crops, peppers and tomatoes and eggplant, in January and February at the latest, and we're definitely cranking them out on the mats. But and there's fancy ones and basic ones. I'll show you a picture of a mat later on. But anyway, you need to pay attention to temperature, and you can always go a cheaper route and use towels to put your trays on with some kind of cardboard or solid surface under that. Okay. What are the ideal characteristics for germination medium? I hope I'm not talking too fast. I've pressured to get through this. So ideal germination medium should be fine, uniform in texture, but well aerated and loose. Free of insects, disease organisms, and weed seeds, and able to hold moisture, but also to be able to drain well. So for beginners, we just honestly, for anyone, the easiest way to go, very reliable, very inexpensive, is to purchase a commercial seed starter. And that's what it's called. You'll be buying a bag of seed starter. Different companies use that terminology. Then you can avoid having to buy a bunch of individual components and mixing your own, which is totally fine to do. But if you're starting out, you don't want to be buying vermiculite and perlite and peat moss or coir and sometimes sand. And there's a couple other components that sometimes get thrown in there. So the key to a good seed starter is it has all of these listed attributes, and it's a peace of mind control to not have to worry about sterility, I think, of the mix. So what it should not be is actual potting soil or soil from your garden or from a flower pot that, where the flower died and you were like, I can reuse this. No, please don't do that. For besides being non-sterile, soils will actually usually be too heavy. They'll keep the oxygen from getting the needed oxygen from getting to the seed. And sometimes it's pretty easy for it to get overly waterlogged as well for optimal rooting. And so the seed doesn't need any additional nutrients in their germination stage. So seed starter it is. Where should you plant your seeds? So I don't want to go on and on about this, but optimal temperature, if you're planting straight into the ground, which is fine to do when the ground is warm enough, the ground needs to be 55 degrees or more reliably at the coldest part of the 24-hour day. So that's not for a long time now. If you can just track your temperature on your phone. And for the purposes of this class, we're talking about starting seeds in containers. You've got a lot of options for your containers. I have pictured here plug trays because I know a lot of home gardeners who seed a small summer garden think that this is the kind of thing you'd only be using for a plant sale or in a nursery or for someone with a ginormous orchard and garden. But actually, you could, if you have a small garden, you could do your entire summer crop just with one of these trays. They're less than $3, incredibly standardized in terms of size for bottom trays, for covers, for heating mats, and you could just you don't have to fill it if you don't need it all. So I don't know, I'm a recent convert to these. You can see they're the same size, but they have a different number of cells. So the one in front has 72 cells and they're maybe an inch square. The other one has over 100 cells and would be for smaller seeds that have tinier seedlings. They're available easily at good nurseries or feed stores, so not hard to find at all. I flipped one of them over so you can see the bottom. This is really important. The chopstick that Diane was referring to is my favorite tool for seeding, and I wouldn't be turning a seeded tray over, but I'd be coming in from the bottom. But you can imagine when it's ready to be planted, I could just pop through the bottom with the chopstick and pop that baby out of there. It's very, very convenient. So instead of going on and on about this, let me just mention that there are plenty of ways of seeding, tons of options. There's 6-packs, there's 9-packs, there's 12-packs, and all of those are fine. We do recommend something with rows so you can keep track with labels. Thanks for doing the heavy lifting on that, Diane, in terms of the importance of labels, because if you think you can't remember what color the cutting is, you're really not going to remember what seed you seeded after you've covered it up. So the tags need to go in first. And the absolute essential is that there has to be bottom drainage. So holes or slits or something like that. So, okay, let's talk about the actual seeding process here. Some tips for successful seeding are proper moisture and compression of the medium before seeding, vital seed packet info, knowing how to glean that from the packet, slow seeding, proper watering after sowing, and proper temperature and light. And we're going to look at each of these tips separately. So proper moisture and compression. I'm going to demonstrate this in 5 seconds for you on Saturday. So again, hoping the wording makes sense here. You'll mix your seed starter with a minimal amount of water. It should still be loose, but not fly away dry. I know the word that Diane used was fluffy, so I'll use that again too, because light and fluffy is what you want. You'll fill the tray or container. You will not press down, but you will take on a firm surface, a solid surface, take the tray or the container and firmly tap it on the surface. And that'll take care of getting out the air pockets, but it will keep it still light and fluffy. Then you're going to, you'll see after tapping it that the level goes down in certain areas, that it's not, you haven't, filled it evenly, and then you'll tap it again just to even things out as you put a little bit more seed starter on top. And then you're going to seed and cover according to specifications, and we'll get to that next. The seed packet is your friend. So you can see in this photo, I've put in 3 different companies. The wording, the formatting is slightly different, but the info that you need for seeding is there on all of them. So, the timing for the seeding, the depth of sowing, and the estimated days for emergence. And if you'll notice on these, one, it says days to emerge. That's when you can expect to see the sprout. The one to the right says matures in a certain number of days. And then the bottom one says, you know better than this now, days to germinate. But if you can, they're trying to sell to their consumers. So it's the phrasing they decided on for marketing. But that's very, very useful to know. And if you're using heating nets, you can count on the number of days they're being basically cut in half. I want to show you, it goes from minimal amount of info, which would be probably the companies of seed packets that you might buy at Dollar Tree, to just way more info than you might ever want to know. So the next slide shows you the inside of botanical interest packets, which is, some people consider overkill. It's pretty amazing. This is on the inside of the packet, so it's really hard to read. You can, you're kind of trying to hold the packet and look in there. I find it a little bit bizarre because you have to, usually I end up with seeds in the packet, and you can't really do that here. You have to figure out what to do with the seeds after you cut it open to read all the stuff. But my point just being that it goes from minimal to maximum info on your seed packet friend, but the basics you're going to find on all of them. Okay, so going slow with seeding, you're going to first make tags for each row. And then for seeds that are large enough, I can't emphasize enough first that the importance of slow seeding, or the total control that your fingers and your brain have over the process. No matter how tedious it might seem, you're going to thank yourself later. So you remember the differences in size of sizes from the seeds from that earlier slide. So as long as you can pick up the seed individually, which you can do for broccoli and kale and those smaller ones, as well as like ones like this that are squash, some kind of squash, I think. You want to pour the seeds into the cupped palm of your non-dominant hand, and then place them individually in the center of each cell with the forefinger and thumb of your dominant hand. For seeds that are too tiny, though, to pick up individually, and they do exist, you're going to take a pinch of those seeds from the cupped palm, and then you're going to move down this row of cells very slowly, rubbing your 2 fingers, your forefinger and your thumb together, to release 2 or 3, hopefully not more, seeds in each cell. And you can see them dropping if you're going nice and slow. Okay, there's that. And then the seed packet tells you how deep the covering should be over the seed. And in general, by the way, the covering depth is 2 to 4 times the seed's diameter. But if it's a big seeds like squash, it's just 2 times the diameter. If you're sowing seeds of different sizes in the same container, it's very challenging to accommodate the different depths. And so some people poke holes and then drop the seeds in. I urge you to not do that, because if the medium is truly light and fluffy, who knows where that seed's going to land, how deep it's going to go or not go. And so what I recommend, and this is the only time you're going to hear either of us recommending that something gets pressed into some kind of medium, I recommend that using your forefinger, you lightly press the seed to the depth in the seeding mix that is indicated on the packet. And for tiny seeds, this will just be, basically settling the seed on top of the cell medium. Then it should be so some will be pressed in more than others, right? And then you sprinkle the medium mix consistently over the whole level, just over the container, leveling the top. And that should do it in terms of depth. And then you want to mist immediately to avoid flyaway medium. And then you want to water it in thoroughly. And to do that, you should use a fine spray so you don't disturb the seeds, the ones that are the most shallow. And in addition, because there's usually a significant amount of peat moss or coir in seed starter, you want to water it in and then let it soak in, and then do your fine spray again, watering it. And you can tell when it's starting to retain water because of the heaviness of the tray. Okay, all of these things are much easier to show than to describe. So, and I should mention also that you want to, of course, do that entire seeding process out of the wind, both on behalf of the seeds and the medium. So you want to go super slow with your seeding, or else you're going to have something like this is going to happen. And this is very important to avoid, because it's just a nightmare. The roots of these every root would be compromised, even for the ones you're trying to save. To be pulling this stuff apart takes tons of time and just a huge headache. So, okay, so I've already mentioned the delicate balance of optimal moisture and ideal heat and light. And let me just move a little quickly through this. Misted and moist, but not waterlogged. Strong indirect light, but not direct sun. And consistently warm medium, but only before the seedling emerges. So since we don't all have fully equipped greenhouses with misters and heating coils and control environments and stuff, you're improvising to simulate the conditions that you want. So you can see the evaporation inside that tray cover there for that seeded tray. This is a heating mat that's reasonable in price, that matches the standardized tray size. You'll notice also in the covered tray, it's sitting in a tray. And if you need to, after seedlings emerge, if you can't be misting regularly manually to keep it moist evenly, you can do a little bit of bottom watering, but you just need to be very careful with that. It needs to just be a small amount, which needs to evaporate before you put more in for at least a day or two. I only really recommend bottom watering for seedlings that are so tiny that they'll disappear if you do anything other than mist them until they get a little bit of growth going, or for super spindly little seedlings that are that way even in their normal growth cycle. So the last thing is you want to make sure to take the cover off right. We've talked about that already. And then seedlings will bend incredibly quickly towards the light. So just be prepared for being able to turn your containers around a little bit. If this all sounds super complicated to first timers, you gain confidence every time you do it. And there's just a couple things to remember you want to avoid to not undermine your success. And that would be overwatering, and then it would also be underwatering, and it would be not hot enough, and it would be too warm. And then lastly, just a lack of consistency, uniformity in your watering. So, or the heat or light for the overall container. So those are probably the most common negative issues. So the cotyledon are the first two leaves that pop up. And I've used tomatoes here because I think most people are familiar with what tomato leaves look like. So on the left, there are three pictures here if you haven't noticed. On the left here, you'll have the first two seeds that come up, and then the true leaves emerge in the second leaf, which are shaped more like tomato leaves. And sometimes they'll appear above the cotyledon, sometimes they'll come as part of the intersection of the cotyledon there. And then, the continued growth happens. So the point here being, you need to wait for the true leaves to appear before planting germinated and sprouted little seedling. But then once the minute the true leaves appear, that's your trigger, and you better get cracking there and pop that baby out and plant it as soon as possible, because the true leaves indicate to you that the roots are developing quickly. They're ready to go. They can use some nutrients from potting soil, and they want to expand. So lastly, we're just going to talk a bit about removing the plant start from the seeding medium. So sometimes the whole plug will pop out with that chopstick method, but often and more commonly, it'll look more like this when you pull it out. So I probably shouldn't use the term pulling. We want to avoid that. So the entire focus needs to be protecting the roots and keep getting it out intact, the whole thing. So if it means jiggling things around a little bit with that chopstick to make sure things are loosened, people also will come in with a little spoon from the top, or there's a method to use a chopstick from the top of the plug or the container as well. The point being that you need to just pay attention to being very closely watching over the roots. So as you loosen it, and then, feeling, is it giving way, and can I get this out? And you want to hold it right below the true leaves, like you'll see in that picture there with the gloves on the hand. And then to plant it, we'll just bring this baby home. You do want to ignore most of what your grandma told you. I was telling Diane, I noticed that both her sister and my sister are on this right now, listening to us. So I do not mean to be casting shade on our moms and grandmas, but there should be no pressing involved. There was a lot of pressing involved with my mom with planting. She'd press as much soil as possible into the container. She'd press a hole into she'd press the plant in, she'd press the soil on the top. We're gonna move away from that. And the relearning is you put a small amount of nutrient-rich potting soil, to use Diane's phrase, in the bottom of the container, and then you hold this seedling and you move it right over, you center it to the container and move it down to the height that you'll want it to be. And then using your dominant hand while you're still holding the seedling there, you start dropping potting soil into the container. And you just keep doing that until it's up high enough to where you want the plant. Then you do a light tap to get the most obvious or problematic air bubbles out. And if you need to add a little bit more around the surface, but you are not pressing or doing any kind of other moving around of the plant, and hopefully you haven't touched the soil. If the plug comes out intact, You do not have to separate the roots, another grandma issue. The roots will know what to do. You just place it on the soil and then put up more soil to level it out to where it should be. And lastly, you want to nurture those first plantings. Just think human baby parallels. I think they've been given their first food there, the nutrients in the potting soil, but maybe they'd like to still be misted a little bit, and they should stay in their same environment for a bit. And the planting medium matters. You want to make sure it's light and fluffy still, and has it's sterile, and the containers are sterile, and it has good drainage and all the good caveats. And then when your plants are sturdy enough to start thinking about planting outside, and the temperature is warm enough in the outside, you want to just do a process that's called hardening, which is basically gradually slowing the plant growth so that it can withstand the changes that it's going to experience when it goes outside from indoors. And so you want to allow 2 weeks for that, and put your plants outside in the shade always for a couple hours a day, and you increase the number of hours over the 2 weeks. And then if the temperature is suitable, you can go ahead and plant it. So seeding, what to expect on Saturday. We will have available for you 6 packs and seeds to choose from, and you'll be able to prepare 2 6-packs with medium for seeding, and we'll have a variety of seeds. You'll practice your labels, seeding the medium, watering it in, all of it. And then we have enough material inventory in the greenhouse for you to be able to practice planting a seedling too, even though that we're kind of, moving it forward there for you. We don't want to just ditch you with just the seedings, but we want you to feel confident when your seedlings appear. And if you have a chopstick that you don't mind sticking in the dirt, please bring it. But otherwise, the deal is the same. Prepare to have fun. And I'm sorry if I have gone on too long. I think my shtick is done.
Great job too. Great job. A lot of wonderful detail. So now let's look at the questions. Do you want to go ahead?
Okay, I was going to say we'll go ahead and just go through what we've gotten to this point. Let's see. Do you ever use a rooting gel or powder to stimulate the root growth?
Diane, you're muted.
Yes, you would on certain cuttings that are sometimes semi-hardwood, definitely hardwood, and some softwoods you would use. There's different potencies of rooting hormone that you would select for different plants that different stems that may not root as fast as another stem.
So in other words, if we stay with the herbaceous at this point, it shouldn't require a rooting hormone.
Right, that's right. Yeah.
Okay. All right. Let's see. Oh, so a question about vegetative propagation. I've had luck starting herbaceous cuttings in water with the fish bubbler providing extra O2. Is there a reason not to do that?
No, that sounds okay. I mean, if you're having success with it, then keep doing it.
Yeah, water is fun too because you can see what's happening.
Yeah, definitely.
Yeah. All right. Do you happen to have a recipe for a nice fluffy seed starting mix in case we wanted to make our own?
We kind of do. Yes, we do. Our proprietary mix at the greenhouse. We have a recipe. You want me to talk? You want me to say what it is?
Well, she's asking about seed starter though, not potting soil.
Okay.
So
there you go.
There's so many different ones, but they just vary a little bit with sort of the basic same elements, right? Vermiculite, perlite, some kind of water retainer, coconut coir, peat moss, and sometimes people put in sand. But the formulas, you just have to go online and look at that. We're not really allowed to sort of advocate for certain companies over others.
We could ask Delise, who asked the question, for her own, because she's an incredible seed starter as well. Delise, you want to share, unmute and share your own recipe? Yeah, maybe she had to drop out.
Oh.
No, she's there.
You busted me.
Oh.
You outed me, Trink. No, I thought the prop group had a you talk about purchasing it. It's very expensive when you purchase it, and it's about 3 ingredients. And I just think that there's a lot of them out there. I'm very lazy, and I have a very lazy way of making my own seed starting mix that I don't measure anything. I just sort of throw some potting soil in with some coconut coir and call it a day, and that usually works for me. But I thought you guys might have something that you would be willing to share that I could learn from.
I think Diane and Charlene's issue is also just making sure that it's sterile, and really protecting those seeds by the skin.
And I would argue that nothing in a bag that you get at a store is going to be sterile inside. I mean, there's more and less sterile, but it's got holes in the bags. It gets roughed around, and I've had many professionals tell me there is not really sterile seed mix. There's soilless seed mix. That's less likely to harbor bacteria and fungi.
These are all good points for those of you that are just starting it. I know when I first started, I really liked just having the bag because it was a small quantity that I knew I wasn't going to use again. And so, you just kind of weigh it all out. There's lots of recipes online. And if you want to create a larger amount, and so you have to buy quantities and put them together, so you'll end up with more. So.
And we are happy to show you what we use at the greenhouse for both seed starting and potting soil on Saturday.
Great. Thanks, Delise. Okay.
Right. Okay. I did put the link in just a minute ago into the chat where you can register for Saturday's class if you'd like. By the way, in case you're not watching that as we go along yet. All right. Let's see. Where are we here? Planting peas and beans. Do you recommend soaking the seed overnight first?
Diane, you want me to take this?
Yeah. Go ahead.
Well, no, I don't. Unless it's some kind of bean I'm not familiar with. Beans and peas are 2 of the easiest things to, work their magic on their own. There's nothing wrong with soaking them, but they're pretty easy to grow on, just as long as the moisture, as long as the medium doesn't dry out. So.
And I would say lupin, there is a scarification process for those. But you can also put those in wet paper towels, and it'll break the dormancy. It'll come through the shell on that. So I wouldn't put it in water, but I'd put it in damp towels, paper towels.
Okay. All right. Let's see. I'm going to go ahead and just read some of these straight out. I've seen some seed sowing demos done by propagators who really do pack their seed containers before sowing. Not like stuffing a sleeping bag packing, but definitely tamping down. They have said it is to ensure the seed to soil contact. What do you think?
Yeah. I think those are strategies. This is just my opinion, but I think those, from what I've observed, and I do work up at the ARB on Tuesday mornings. From what I've observed, those would be the kind of strategies that are being used for mass production. And what we're talking about here is going slow for a small amount of seeding that you are in control and you're seeding for yourself, for your own purposes. And if you have to seed 50 trays a day, I think things are going to be totally different. So that's my reference to that. What do you think, Diane?
Oh. Sorry, my phone went off, so I cut myself off. I said that sounds about right. Yeah, okay. Definitely.
Mm-hmm.
All right. So expanding a bit on Charlene's comment about artificial light producing leggy seedlings, with broccoli seedlings in particular. Can you expand a little bit more on that? And also, once the seedlings appear, do you turn off the heat mats?
Yes. Yes, thank you. I love that question. Overly leggy plants there's a ton of YouTube videos on that because it's just a huge issue for home gardeners. So you're not alone, whoever asked that question. So backing up, doing your second question first, yes, remove the heat after the seedlings pop up. I would say if it's a very low heat and you know what you're doing with eggplant, peppers, and tomatoes, you might keep it going for a little bit. But in general, the heating mat limits the regular growth pattern. It changes it, not limits, but makes it pop. It definitely. You can attribute legginess to lights and to heat, definitely. So there's a few ways of mitigating legginess in seedlings. So I'll just go ahead and go right to that. One thing you can do is, as soon as your plants start popping up, for things like broccoli, since you mentioned that, or kale, or arugula, or you can take your hand and lightly brush it over the like this. I'm demonstrating here and no one can see, like this, lightly over the seedlings, and it'll toughen them up a little bit and help them get a little bit sturdier. It's a sort of a human manipulation there. And but definitely unplug the heating mat, take those lights off if things are getting leggy. And then when you're planting them, it's very difficult to wait for the true leaves when things are so leggy. You just want to get them sunk into potting soil ASAP, so you don't have to look at that legginess anymore. So it's hard to wait for the true leaves, but if you can just wait it out, then sink that baby when you plant it. Go ahead and bury it. Bury as much of that legginess as you can. Don't be afraid to do that.
All right. That is all the questions. There's one other one about the plant sale, but I'm pretty sure that's going to be answered on this slide right here. So Trink, I'll turn it back over to you and everyone.
So the link that in this slide, and Denise is going to now put it in the chat as well, is a link to an evaluation. And we ask that you give us feedback. We always appreciate the feedback and know how to improve our classes, and give us ideas for other classes that you might like. Also, within the next month or so, you'll get an email survey from the California Master Gardeners Organization. They also like to get feedback on how all their Master Gardeners are doing. It helps them with fundraising if they can hear, how much we're helping the home gardeners. So that helps us as well. So upcoming classes, we have an IPM for early spring pests class that we've done a few times and is always quite popular, about primarily gophers, snails, and slugs and birds. Those things that once you put those seedlings out into the garden may give you some trouble. So we're going to do that on Saturday, April 8th at Quail Hollow Ranch up in Felton. That'll be posted to our website within the next few days. So, and then the Master Gardener Plant Sale, April 17 to 26. And I'm going to let Diane and Charlene tell you about that, since that's their big event. What are you going to be selling, guys?
Vegetables and herbs, lots of native plants, primarily some annuals and perennials, and lots of salvias, and some bedding plants. You want to go on, Charlene?
Oh gosh, some indoors.
Yep. Tree stumps.
I think I just need to say like 22 varieties of tomatoes.
Wow.
And the thing with tomatoes is we try to start them early enough so that people are successful with them, and so they'll be fairly mature. Hopefully the weather is so crazy, but we're doing our best, and we have recommendations according to where people live or their conditions. So if you need to have your tomatoes in containers on your balcony, we can tell you which are best for containers. And if you live in Monterey or Pacific Grove and see fog more often than sun, then we can also help you with that. So.
And the plant sale is also a little unusual in how you order the plants and then pick them up. Will you explain that to people?
Go ahead, Charlie.
Well, we used an online platform, Shopify. And then after you do your purchasing, your shopping and purchasing, which you can't do until the sale starts, till the sale opens, then you sign up for a pickup appointment, and through a second platform called TimeTap, and they're linked. And then we have plant sale pickups at the greenhouse there in Salinas for people that live in that Monterey County and or are going that way for work. And then we have a few days of pickups appointments scheduled at our demonstration garden in Watsonville for Santa Cruz people. So hopefully people won't have to drive too far.
That sounds great. And as I recall, it's a lot of plants go in the first couple days of the sale. So if you're interested in getting particular things, really do get in there the first couple days. Because
6 o'clock in the morning, April 17th.
We usually do sell out of our most popular natives and some unusual salvias, and I don't know, I can't. It's shocking. The 90% of our sales happen in the first 2 days. So
great. It's really great. Well, thank you two very much. I want to thank you so much for doing this, particularly while you're working so hard in the creating plants for the plant sale. So thank you for taking the time. I hope questions were answered about Saturday. You can register online for that. And then you'll get the information about where the plant sale is. And we always finish with this slide just to remind everybody that we do have what we call a hotline that's on our Master Gardener website. You can take a picture of something you're having problems with and send it in to us through the hotline with the pictures, and you'll get an answer within no more than 5 days, so usually 2 to 3 days. So I hope anytime you get stuck on something, be sure to let us know. So thank you again, everybody, for coming. Anything else you want to add, Denise, Charlene? Just go out and plant, huh?
Yeah, just a great class, and I hope we'll see some of you on Saturday. It's I learned a lot too, so I can't wait to get out there and start some little seedlings going here. So. All right.
Thank you all very much.
Thank you.
Thanks.